Good morning, guys, and welcome back to Excelsior Academy. So today we are going to do the column chromatography, and there are three types of column chromatography, namely ion exchange, size exclusion, and affinity chromatography, of which we are going to study today ion exchange chromatography. Okay, so it is the most powerful methods for fractionating proteins which make use of this column chromatography. Now it is based on protein charge, size, and binding affinity. So based on the protein charge, we can have either positive charge or negative charge of the proteins. Then size, whether it is a larger protein molecule or smaller protein molecule, right? And then based on the binding affinity. Now based on these factors, there are three chromatography present. Okay, so based on the charge, that is plus and minus, it is called ion exchange, which is today we are going to discuss about. Then based on the size, we have got gel permeation chromatography, which is also called size exclusion chromatography. And then we have, based on the affinity, it is called affinity chromatography. Okay, so we have got three chromatography which we will be completing in three coming lectures. Now among all these, so this is based on the separation is based on the either the charge or size or the binding affinity of the proteins. Now something is there which is common to both, and that is the stationary phase and the mobile phase. Now to understand what is stationary phase and mobile phase, you have to imagine there is a column. Okay, so this column is made and you have got the stand which is holding the column. Then this column is either made up of borosil, glass, or polystyrene. Okay, so these have to be inert. What does it mean? It cannot react with the matrix or the protein It cannot react. Okay, so the surface shouldn't react with the matrix or the protein. Now this column is filled with the matrix. So this will be filled with the matrix. So different types of matrix are there, like Cephadics. We have got the agaros also. So all different types of solid matrix is there, right? Now, what happens is once you prepare your protein extract, remember in the previous lecture we talked about separation of proteins. So you do the cell lysis of the cell, let it be bacterial, plant, or animal, you do the cell lysis. Centrifuging and supernatant you get. After supernatant, what you get is called crude extract preparation. Then you go for fractionation, that is ammonium sulfate precipitation or pH precipitation. Then you dialyze against the required buffer. Now what you get is your protein solution which is ready for chromatographic techniques. Take that protein solution and add over here. Okay, so with the help of the pipette you are going to add over here. So it has to be done very nicely so that uniformly it is present on the top. Then what you do is, then on the top you start adding the buffer. Okay, so now the buffer, this buffer is going to be the same in which you have dialyzed your protein. Remember, you cannot just change the buffer anytime. Always keep the same buffer. Now this is a buffer against which you have dialyzed your protein. Same buffer you will be taking once this protein starts going inside. So this protein will start going inside the matrix uniformly, okay, depending on the size, depending on the charge, or depending on the affinity. And then you keep on adding your buffer. Okay, then what happens, this buffer is called your mobile phase. It is called your mobile phase. Okay, so this is your stationary phase and the buffer which you add on the top is your mobile phase. Now, the protein containing solution layered on the top of the column percolates through the solid matrix as an ever expanding band within the larger mobile field. So what is happening? Depending on its property, slowly it will go down into the matrix. Okay. Individual proteins migrate faster or more slowly through the column depending on their properties. So just imagine, let us talk about one example. You have got the column, okay, and then pore size are there. Now, if you have got a larger protein, what will happen? It will not go inside the matrix. It will present outside the matrix. And when you start adding the buffer into this, what will happen? This larger protein molecules will just go from outside of the matrix and will come out. Okay. So it means that larger the protein molecules, early will be the elution of that particular protein. Okay. Now, once you start eluting out these proteins, you collect into the glass test tubes or in the appendose. Now, each appendose will be collected and visualized under the spectrophotometer. Okay. Now, as I mentioned before, the fluorescence of each protein is because of the 3 aromatic amino acids. What are those? Phenylalanine, tyrosine, and tryptophan. And this gives absorbance at 280 nanometer. So this reading will be taken in the spectrophotometer, and wherever you get the readings like more than 0.1, you select that particular appendose, and rest all, if the readings is near the blank, you will discard. Okay. So this is how you carry out your protein purification. So let's talk about ion exchange chromatography over here. So ion exchange is a chromatography process that separates ions and polar molecules based on their affinity to the ion exchanger. So from the name itself, it's clear that it is going to exchange the ions. Now, what kind of ions are there? Cations and anions are there. So it means it is going to exchange for its cation or anion. So it must be done in conditions that are 1 unit away from the isoelectric point of a protein. Now, why this point is important? That it has to be 1 unit away from the isoelectric point of the protein, because we know that at isoelectric point of protein pi, the protein has no net charge, right? No net charge. So what happens when there is no charge to the protein? It just precipitates out. So what will happen? You are not able to proceed with your protein purification. Okay. Yes. So this is how the latest or the modern ion chromatography system looks like. And usually when I will be discussing with the conventional method so that the principle is clear. Nowadays we have got this modern ion chromatography system in which, see, if you see over here, you have got everything automated over here. So you have got the screen and you can see over here some spectrum is given, right? So wherever the spectrum or the absorbance readings are coming, only those appendose or the test tubes you are going to collect. Okay, wherever the readings is flat, there you cannot have the there you are not going to collect the sample. Okay, now then this is your machine. You can see over here the buffer is kept. Now the buffer is going to act as your mobile phase. Okay, and inside itself you have got your column also. So inside itself the column is present. In this chamber your column is present. So usually when we do the conventional, we keep our column straight, whereas in this case when you have got the machine automated, you keep it your column as in the horizontal position. Then what happens? It goes, and from this pipe it goes over here, and each test tube stands are present over here in which automatically it gets collected. So you program in such a way that wherever the peaks are coming, then only it is going to collect, and it rotates and collect the sample. Okay, and whenever there is flat, it is not going to collect, because flat means the absence of protein. So now what you can do is, you can have wherever the peaks are coming, only that time you can collect the sample. So from this, you don't have to go to the spectrophotometer because it is attached to it. And what you can do is, wherever it is coming, you can proceed. So only the test tubes having the reading will be collected. So in this way, your most of the job has been done. So there are two types are there, anion exchange and the cation exchange. So we have got two types of ions, right? So we have over here anion exchange and the cation exchange. Now in the case of anion exchange, stationary phase is positively charged and negatively charged molecules are loaded to be attracted to it. So what does it mean? It means that in the column, whatever matrix is present, that matrix is positively charged. Okay. Now your proteins needs to be negative charge. So here your protein should be negative charge. Okay. So what will happen? So when you pass through the sample over here, what will happen? Wherever the positively charged matrix is there, it will come as and it will bind, having the ionic interactions. Okay. Now in the case of cation, so it is just the opposite. So here the stationary phase is going to be negative charge and your proteins are supposed to be positively charged to be attracted to it. Okay. So we have got anion exchanger and cation exchanger. So you need to know what kind of proteins are there you want to separate. Okay. Now talking about the column resolution, it means what? In the same way we talk about the microscope and magnification, right? So we need to talk about the column resolution also. So resolution means how nicely two different types of proteins have been separated, right? In the same way, in the microscope also we see that how nicely or how distinctively the two objects have been separated so that we can see the both objects clearly and not as the merged one. In the same way, we also want our proteins to be separated very distinctively. Okay. So for this, we are going to talk about the column resolution, how to improve the resolution of the column. So first point is that as the length of the column increases, the resolution of two proteins or two types of proteins with having different net charges improves. So if you have got a 10 centimeter column or a 20 centimeter column, so which one is going to give you the better resolution? It is your longer column. Okay. The rate at which the protein solution can flow through the column usually decreases with column length. So what is happening? Increase in length, flow rate will decrease. Okay. Now how slowly it moves and all, it depends, because if it is very slow, what will happen? It is going to get diffused. Okay. It just spreads and there is no separation. But if it is in the appropriate flow rate, then nicely it will get separated on a lengthy column. The length of time spent on the column increases. The resolution can decline as a result of diffusional spreading within each protein band. So this is what I was talking about. See, if you have got the longer column, okay, if the flow rate is good, then it's well and good. But if you keep the flow rate very slow, what happens is slowly, different proteins, let us say that this is protein and this is another protein. Okay. So it is moving according to their molecular mass. So it is moving. Now, if you keep the flow rate so slow that the proteins are moving very slowly, what happens is they try to get merged. So because of which, what will happen? They get mixed. So here it gets diffused and it starts spreading. Okay. So what will happen? Finally, I am not going to get the separate proteins. In my test tube sample, I'll have both the proteins, which is not required, right? Because I'm doing the separation chromatographic technique. I don't want to mix up my proteins. I hope this is clear. Now, this is how your cation exchange chromatography is. So you have got the polymer beads with negatively charged functional groups, right? So we are going to exchange cations, that is positive charge. So your matrix is going to have the negative charge like this. So there are beads onto which we have got the negatively charged ions. For example, sulfonic acids. For example, sulfonic acids. So they give the negative charge to the solid matrix or your stationary phase. Now what you do is you add your sample. Okay. So your sample is added onto the column. Then once it starts going inside the matrix, you add your reservoir. So this is your reservoir, the mobile phase. Okay. where this matrix is your stationary phase. Then slowly you start collecting the sample. Okay. Now in this case, see, so proteins move through the column at rates determined by their net charge at the pH. So what will happen? Once you start adding over here, the negative charge are bound with the positive charge proteins, right? With cation exchangers, proteins with more negative charge move faster and get elute earlier. So what does it mean? If you have got negative charge protein and we have matrix also negative charge, so what does it mean? The interaction is going to be weak, correct? So when I add buffer, what happens? This is going to elute out fast and first also. Is it clear? Now if the sample or the protein is positively charged, so it will be bound to the negative matrix, and hence this is going to be very strong. So I need strong pH or salt in order to break this ionic interaction, and hence what happens? It will be eluted out last. Okay, so if you have got the negatively charged proteins, though, those will be eluted out first, and if you have positive charge, then it will be eluted out last. Okay, and just the opposite case in the case of anion exchanger chromatography. Yes, so it will be more clear over here. So a mixture of proteins, in this we have used the MES buffer. MES, MES buffer. Okay, so it is loaded onto the cation exchanger. So positively charged proteins will be adsorbed to the media, displacing your cations. So you can see the matrix is over here, negative charge. Okay, and then you have got the positive charge. Now a pulse of MES is formed. To avoid drastic ionic strength increase, a pH decrease in column microenvironments, applied the concentration of adsorbing protein should be this much. So what they're saying is that, see, if you see the figure, we have got our carboxymethyl anion. This is your solid matrix. Okay, then you have got the positive ions, cations are present. Now what you do is you start adding your sample. Okay, so this is your sample, and the sample has got the MES anion and the protein which is having the charge, positive charge. If you see, this is your protein of interest having the positive charge, and it is mixed with the MES anion, that is in the buffer. So it is used, the MES buffer is used. So what you have done, you have got the proteins, okay, and it has been dialyzed against the MES buffer, and MES has got the chloride anion. Now what happens is you have added this protein solution into the column. Okay, so what will happen? The cations are getting exchanged. It means instead of Na plus, now your positive charge proteins are getting bound to the matrix. Okay, so what will be eluted out? In the buffer, you have only your MES anion, this one, and the sodium ions. Getting the point? So in the column, you have got the negatively charged solid matrix. It has been equilibrated with the buffer, and they are sodium cations are present. Now you have your protein to be loaded, which has been dialyzed in the MES buffer, and MES buffer has got your chloride ions. Okay, now once you start adding, what happens is cations are getting exchanged, and that's why it is called cation exchanger. So instead of Na plus, now your positively charged protein will get bound to the negative charge solid matrix, and in the elution you get your Na plus and Cl minus. Okay, now this is there. So we have got the positively charged protein over here. So what will I do? Depending on the proteins, the positive charge varies. So you can see that There are proteins having one single positive charge or two positive charge or three or many. So what we do is we know that the salt disrupts the ionic interactions. Okay. So what we do is we start eluting out the proteins with increasing salt gradient. That is starting, we use low molar concentration of the NaCl. So let us say that we have used 400 millimolar NaCl. So in the buffer we add NaCl. So what will happen? The weakest interaction will start coming out. So once this kind of protein having single positive charge is eluted out, you get a peak over here. Okay. And then nothing is there. So when nothing is coming, you see that now no proteins have been eluted out. What you do is you increase the salt concentration. Okay. Then what happens? More positive charge proteins will be getting eluted out. So you get another peak. Similarly, at the end, what we do is final concentration we use, and that is around 1 molar NaCl. So we don't go beyond this. So 1 molar NaCl is the last concentration of the salt which is used, and finally you get all the proteins. Okay. Now this you have checked in this spectrophotometer at the 280 nanometer. Now however, how many test tubes or appenders are present, you're going to take the reading. So you get graphs like this. Okay. Now again, to check whether these proteins are pure or not, you carry out SDS page to check the protein purity. Okay. Don't worry, we'll discuss this also. So I hope this was all clear. Now let's talk about protein elution, which I have already mentioned, but just in more detail. So the concentration of the exchangeable counter ions which competes with the molecules for binding can be increased or the pH can be changed. So this is what we were talking about, that when there is a plus and negative charge, we need to break this interaction. So this can be done either increasing or decreasing the pH or by increasing the salt concentration. Okay. Now a change in pH affects the charge on the particular molecules and therefore it alters the binding. So increase in pH and increase in pH or decrease in pH will alter this interaction and hence it can be eluted out. But basically in the lab, like for example, for my case also, I have done ion exchange chromatography. So I have done anion exchange chromatography and I have always used salt gradient only to elute out my protein. Now the molecules then start eluting out based on the changes in their charges from the adjustment. And finally, it releases the protein of interest. Now, how do you understand that your protein of interest has been released or eluted out? Just by carrying out the SDS page. Okay. Now, sometimes there are few people who can also go for the enzymatic activity. If your protein is an enzyme, you can just take few microliter from your test tube and just carry out the protein activity. Okay. So this will also confirm that in that particular test tube, your protein is present. On the other hand, step elution can be used in which concentration of counter ions are varied in one step. So that is the gradient wise, you keep on changing. Okay. Yes. So when cation exchange chromatography is used, cations will elute out last, right? Because you have got the negative charge support and the positive charge protein. So more the positive charge protein, last it will come. If it has got less positive, it will come earlier. And if it is negatively charged protein, then it will come the first. Because negative will not find any attraction and they will elute out first. So same thing, the negative charge molecules will elute out first. Limitation to this purification technique is that it is limited to ionizable group only. So if you want to purify your proteins, which are like based on the size or the affinity, then you cannot use this chromatography. In this case, only the positive or negatively charged proteins can only be separated. Now, what is the principle behind this? So this is nothing but what already have been discussed. So in case of cation exchange chromatography, it retains positively charged cations because your stationary phase is having negative charge. So let us say that this is your stationary phase which has got the negative charge and a cation has been attracted. Remember, in that case we have got the Na plus as your cations. Now this is your protein and it is present in some buffer which is having the anion. Okay. So in the previously discussed, we have got the chloride. Now what happens? In this case, there is an exchange of the cations. Okay. And these two ions, that is Cl plus, C plus and B minus, are eluted in the buffer. Okay. Now to get the M plus, which is your positively charged protein, either you go for pH gradient or you go for salt gradient. Okay. Similar case with the anion exchange also. So here you have got the positively charged solid support, that is a stationary phase, and a chloride ion for the exchange. Here your proteins is negatively charged. Okay. So your protein goes and binds to the matrix and these are eluted out first. And finally, with the help of the pH gradient or the salt gradient, you will elute out your protein. Okay. Simple. Now, what's the procedure? So just let me tell you once. You have got the column. Okay. So in the column, you wash the column. It's empty column. First, then you have to make the slurry of your matrix. Okay. Slurry means you're going to equilibrate the column with the buffer in which you're going to elute out your proteins. Make the slurry. Start adding into the column. So on the bottom, you have got the glass fibers. Okay. So the glass fibers will not allow your matrix to pass through, but only allow the proteins and the buffer to pass through. Like only the liquid can pass through and the solid matrix will not pass through. Now you need to equilibrate the column. So with the buffer, you keep on adding till the matrix becomes of that particular pH in which the protein you have dialyzed. Okay, so you cannot change the pH here and there. Now what you do is, let's see what is written over here also. So once equilibrated, the charged ions in the stationary phase will be attached to its oppositely charged exchangeable ions like chloride or Na+. Okay, so this depends on the buffer you use for equilibration. Now, buffer choice depends on the proteins in which it can bind. So after equilibration, the column needs to be washed. So wash means, see, when you're making all this preparation of the slurry and putting into the column, there are chances that impurities will be present. And I don't want that impurities to come into the protein. So what will I do? I'll keep on washing till all the impurities are washed away. Then protein loading. So as mentioned before, you have got the column. Okay. And now it is filled with the matrix, equilibrated with the buffer. Now I'm going to add my protein. Okay, so I'm adding my protein. So slowly what will happen? It will go down depending on its property. Okay. So the sample buffer needs to have the same pH as the buffer used for equilibration to help the desired proteins to bind. Now, so we have already done the column washing. Now after to carry out the protein elution, what you can do is, so in the figure as shown before, so your proteins are there which starts going down, right? Now it is still here only. I need my proteins to go down and get separated. So what will I do? I'll start adding the buffer, the mobile phase. So they will also start. Now because of that pressure, the proteins, depending on their activity, either the charges, it will keep on binding, and if there is no charge, it will come out with the buffer. Okay. Now I need my protein which is bound to the column. So what will I do? I'll start adding the NaCl. Okay. So maybe first I can use this 100 millimolar. Then I can increase to 250 millimolar and then 500, and last I'll use is 1 molar. So this choice of gradient of NaCl you can make your own. So initially if you're doing for the first time, then start with 50 millimolar, 100, 200, 300, 400 like that. Okay, so it's like each step wise you're increasing the concentration gradient. Now once you do one time column chromatography, now you have certain ideas that maybe in 100 millimolar nothing is coming out. So you don't have to use 100. Next time you can use 250 directly. Okay, so this is all comes with the experience. Now, with increasing ionic strength of the buffer, the salt ions will compete with the desired proteins in order to bind on the surface of the medium. So what will happen? So these Na plus will try to bind over here, leaving our protein to elute. Okay, so this is how salt is helping us in elution of the proteins. So this will cause your desired proteins to be eluted out of the columns. Now, if the protein have low net charge, it will be eluted out first. If it is having high net charge, then it will need high ionic strength for them to be eluted out of the column. So they will be elute out in the last. So what are the types of stationary phase? I have mentioned already few. So examples are trimethyl aminoethyl, triethyl aminoethyl, diethyl to hydroxypropyl aminoethyl, aminoethyl, diethyl aminoethyl. So what I have used for my experiments mainly is DEAE, that is diethyl aminoethyl, and sulfomethyl, carboxy and carboxymethyl. So they are the anion exchangers. Okay. So DEA is your anion exchanger, and these are your cation. Okay. Yes. Now column packing, this is really a very important step. So as I mentioned before, you have got the empty column, put the glass wool over there, rinse the column with the same buffer in which you have made the slurry. Now what do you mean by slurry? So the matrix are like powder kind of thing. Okay, so they are not soluble in water. It's not soluble, but it will get wet. Okay, now this you will put into the buffer. It will become a colloidal solution. Now to this solution you take and put it into the column. So because of the presence of the glass wool at the bottom, let me show you with the figure. So you have got the column. Okay, now over here we add glass wool. Don't add cotton because cotton will start getting spoil. It will catch the bacterial contamination and all. So we use glass wool. So we'll keep it over here. Now when you start adding the matrix over here, what happens? Only the buffer comes out. Okay, and not the matrix. If the matrix is coming out, it means the glass wool is not inserted properly. So this is how you do your packing. Now there are certain problems are there. Inefficient dry packing means that you are not putting the buffer continuously. And one thing is that every time keep buffer extra so that it doesn't get dry. Because if it gets dry, it forms the cracks in between. And if there are cracks in between, then protein separation will not take place properly. Okay. And you cannot have wet also, because if it is wet, then the protein diffusion will take place, because of which separation will be difficult. Okay, so you have to keep in mind, and with practice it's easy to do. Now cellulose based medium can be used for the separation of large molecules, as they contain the large pores. Okay, now protein binding in this medium is high and has low hydrophobic character. So you need to decide like for what kind of proteins, what kind of matrix is needed. So sometimes it is given in the manual and it is known also. So you have got DEAE as an anion exchanger mentioned before, and then you have got the SAPADX as a positive group having. Okay. Now the ability of the medium to swell in liquid is based on the cross-linking of these substance. So what you do is this, it comes out as a powder. So you put into the buffer overnight and next day you put into the column. So optimum conditions is temperature. So we know that the proteins are fragile and at high temperature they might get denatured. So we keep the columns in the cold condition. So if you have to carry out the protein purification, you need to keep AC on, or you have to sit into the cold room to carry out your protein purification. As well as the pressure. Now we talk about pressure. If you're doing with the conventional method, there is no need to check for the pressure, but if you're using automated machine, then you have to check for the pressure. So it will also determine the flow rate. Otherwise, in the conventional method, it is because of the gravity. So it depends, the flow rate depends on how nicely, because of the gravity, the proteins are getting eluted out. Now, incorporation of high temperature and pressure allows a significant increase in the efficiency of ion chromatography along with decrease in time. So if you're doing with the automated machine, that is the modern ion exchange chromatography, then you can actually increase the temperature and pressure, and which will also decrease the time taken to carry out the chromatography. So if you do with the conventional method, it might take one complete day to carry out, but if you do in the machine, it takes hardly 10 minutes. So these are the differences. Now there is one more thing, the weak and the strong ion exchangers. So a strong ion exchanger will not lose the charge on its matrix once the column is equilibrated, and a wide range of pH buffers can be used. So that is in the case, for example, Q Cephorose is the example of strong ion exchanger. Okay. So in this case is that I can use pH 4 to 10. So because of this change in the pH, the ion exchanger, it is not losing its charge from its matrix. So it is a strong ion exchanger and I can use for wide range of proteins. Whereas weak ion exchangers have range of pH values in which they will maintain their charge. Now, if the pH of the buffer used for a weak ion exchange column goes out of the capacity range of the matrix, the column will lose its charge distribution and hence you will lose your protein. Okay. So if suppose the column is used again and again, so the charges, like for example, negative charge solid phase is there, stationary phase is there. If it loses its charge, what happens? Your proteins are not going to bind, and hence when you wash the column, it will just go into the wash. So these are the examples of Q, which is showing the strong ion exchanges or weak ion exchanges. So as I mentioned before, Q Cephorose is strong. Sorry. So Q Cephorose is strong and DEAE cellulose is in a weak anion exchanger. So just go through these slides and please try to remember few of them so that it becomes easy for you to carry out the big questions to solve. So I hope this is clear to everyone. Thank you for listening. If any doubts are there, please comment us, or you can ask me personally on Excelsior Academy 1228@gmail.com. So thank you for watching. If you like the video, please like, subscribe and share. Let your friends also know, and let us join together to clear our exams. So in the upcoming video, we will be talking about the size exclusion chromatography. Thank you, people.